Hello everyone. Today we are going to learn the topic microbes as single cell protein. Now, many of us are already aware of the term single cell protein. The term single cell protein actually refers to proteins extracted or any microbial biomass which is obtained from unicellular microbes like bacteria, yeast, or algae, or multicellular microbes like filamentous fungi or filamentous algae, which can be used as food supplements for humans as well as animals. Now, SCP is produced when microbes ferment a substrate, which is generally a waste material in case of single cell protein. Thus, the microbes increase in their biomass and in turn SCP is obtained. The term SCP is coined by Carol L. Wilson of Massachusetts Institute of Technology in the year 1966. Now, why we need SCP? When we have our own conventional protein sources, that is the plant and the animal proteins, one may ask the question that why we need SCP. The answer is that proteins are the macronutrients which are the building blocks of living beings and they are required in large amount than any other nutrient. Nowadays, due to the rapid growth in the population, many countries, mainly the developing one, are facing protein scarcity in food. So, it is necessary to produce protein through all possible methods, and at this point, we need SCP because the conventional sources of protein, that is, the animal and plant sources, are not enough to meet this demand. Furthermore, proteins, that is, the single cell proteins, when produced, they use waste materials which leads to environmental cleaning. So this is the way where we can produce food from microbes as well as helps in environmental cleaning. Next we come to the history of single cell protein. Single cell protein remained a part of human diet from ancient time, and before 2500 BC, yeast cells are known to be used in bread and beverage productions. Also, mushrooms were very popular in human diet from ancient time. The Aztec people of Mexico used the algae spirulina in their food. These are the two pictures which shows that mushrooms were used from ancient time in human diet. The first picture shows gold mushroom ornament of head worn by the ancient people, and the second picture shows the underworld deity Soma with mushroom eyes, which is obtained from Mesoamerica and South America. The first industrial production of SCP took place during the First World War in Germany. In 1960s, researchers at British Petroleum produced yeast from N-paraffin, and they called it as proteins from oil. Protein was the first commercially available SCP which was used in animal feed. This is the image of the book written by Frank Matthews Dugan, which deals about the usage of fungi in the ancient world. And the picture on the right hand side shows mushroom stones from Middle America. Next we move on to the production of SCP. SCP production needs two main components. Number one is the substrate and number two is the microorganism. Now, what is the substrate? Substrate is nothing but the media on which the microorganisms grow and increase in their biomass, and thus SCP is produced. Now we come to the microorganisms. For selection of microorganism for SCP production, certain criteria should be kept in mind, among which is that the microorganism should be non-pathogenic to humans, plants and animals. It should have a good nutritional value. It should be produced easily and cheaply in large scale. The microbe should be toxin-free, it should be fast-growing, and it should be easily separable from the media. Next, here is the list of the microorganisms which are used in the production of SCP. two groups have been shown. Number 1 is the algae. Under this group, members of the genera Chlorella and Spirulina are widely used. Under the group yeast, various species like Candida utilis, Lipolytica, Saccharomyces cerevisiae, etc. are widely used for the production of yeast SCP. Now we move on to another two groups, which are fungi. Under this group, the members of the genera Chaetomium, Fusarium and Paecilomyces are widely used, and under the group bacteria, members of the genera Brevibacterium, Methylophilus and Lactobacillus are widely used for the production of bacterial SCP. Now we are going to learn about this microbe in little detail, and we will start with algae. Algae, the genera Chlorella, Spirulina are generally used for the production of algal SCP. The substrate used by these microbes are carbon dioxide and sunlight, which are present in the nature in abundance. These SCP contain about 60% of protein and is suitable for animal feed. Now the disadvantages of algal SCP are that they are rich in chlorophyll and cellulose. The cell density is always low, so the yield is not good. High risk of contamination and the recovery methods are costly. Next we move on to another group, which is yeast. Members of the genera Saccharomyces candida and Torulopsis are widely used for the production of yeast SCP. Yeast SCP is suitable for both human and animal consumption. Yeast SCP is rich in vitamins, mainly vitamin B, and the recovery of yeast SCP is also very easy. But there are certain disadvantages also, which are the slow growth rate of yeast than bacteria. The yeast SCP has a high nucleic acid content, and some yeast SCP are also deficient in sulfur-containing amino acids like methionine, so external supplementation with these amino acids are required. Now we move on to another group, which is fungi, and member of the genera Fusarium, Paecilomyces and Chaetomium are used for the production of fungal SCP. The substrate used by these microbes are starch, wood, sulfite liquor and various other things. The protein content in fungal SCP is around 50%, and the disadvantages are slower growth rate than bacteria and yeast. Contamination is very frequent with yeast, and fungal SCPs also contain a large amount of nucleic acid like yeast SCP. Lastly, the risk of mycotoxin, that is, certain fungal SCP contains mycotoxin, so a thorough evaluation of fungal SCP is required before consumption. Next we move on to another group, which is bacteria, and members of the genera Brevibacterium, Methylophilus and Lactobacillus are used for production of bacterial SCP. Bacteria can be grown on wide variety of substrates. The growth rate are also very fast compared to algae, yeast and filamentous fungi. The SCP contains protein of around 80%. But the disadvantages with bacterial SCP is that the nucleic acid content is very high, which is around 20%. Risk of contamination with pathogenic bacteria is always there. So, a good evaluation with pathogenic bacteria and the presence of endotoxin in the SCP is always required. The recovery of bacterial SCP is somewhat problematic. Now, this is the chart which shows the dry weight percentage of various nutrients present in the SCP of all these organisms. For example, protein percentage in bacteria is high and low in algae. The dry weight percentage of fat is highest in case of filamentous fungi and lowest in case of bacteria. And again, the nucleic acid content is very high in case of bacteria and lowest in case of algae. So from here we can now see that all the organism has their own limitations and advantages. So according to the availability of the substrate, organism, the cost, the microbe should be chosen for the production of SCP. Now we move on to the substrate. As I have already told that for the production of SCP, two main components are required. One is microorganism, which I have already discussed, and now is the substrate. Various substrates are there for the production of SCP, and among those, cellulose is one. Cellulose is present in almost all higher plants, so it is an attractive substrate because of its low cost and abundance, and can be obtained in the form of wood chips, straw, and sawdust. These substrates should be pre-treated thermally and chemically before fermentation, and organisms having cellulolytic enzymes should be used for the fermentation of cellulose. For the example, the fungus Chytomium cellulolyticum is often used for the fermentation of cellulose. This image shows cellulose in the form of wood chips. Now we move on to another substrate, which is whey. Whey is a pale yellow liquid waste of cheese industry, but it contains a large amount of sugar in the form of lactose and various types of proteins. So it serves as a very good substrate. Various yeasts like Candida krusei, Candida intermedia, and bacteria like Lactobacillus are commonly used for the production of SCP from whey. This is the image of whey. Now we move on to the next substrate in the list, which is molasses. Molasses is a viscous, brownish colored byproduct of sugar refining industry. This substrate has a very high sugar content, so serves as a good nutrient for growth of microorganisms. So this substrate can be used for the production of SCP. Molasses also used for the fermentation of alcohol by yeast, from where yeast biomass is obtained and thus can be used as SCP. Yeast like Saccharomyces cerevisiae, Candida utilis can be produced from this substrate. Next we move on to starch. Starch is a substrate which can be obtained from plants, and the fungi Fusarium graminearum can utilize starch for producing SCP. The SCP produced from this fungi has a fibrous meat-like texture which is known as mycoprotein and marketed as meat analogues and is approved for human consumption. Next we move on to industrial effluents. Several industrial effluents can be used as substrate for the production of SCP because they contain large amount of carbohydrates and other organic compounds. For example, sulfite liquor is one such which can be used as SCP production and can be obtained from the wood processing industry. But for using sulfite liquor, first SO2 should be removed from the liquor, the pH of the liquor should be adjusted. Fungus like Paecilomyces variety can utilize sulfite liquor for its growth. Next we move on to another substrate, which is hydrocarbons. Hydrocarbons are obtained from oil or petroleum waste, and many bacteria, actinomycetes, yeast, molds, etc. can utilize these hydrocarbons in the form of crude oil and petroleum waste. Like the yeast Candida lipolytica can utilize hydrocarbon from crude oil. Now, the substrate methanol. Methanol can be obtained from methane. It has a high water solubility and less inflammable than methane, so it serves as a good substrate for the production of SCP. The bacteria Methylophilus methylotrophus can be grown on methanol for the production of SCP. Now we come to the commercial production of SCP. Commercial production of SCP is required to achieve two main things. Number one is that a CP serves as a source of protein for animal and human. Second, it also serves as a source of inoculums in food fermentation industries, agriculture, and waste treatment plants. There are certain factors which affect the production of SCP commercially, which are productivity and selling price. On a large scale basis, when SCP is produced, the production should be high and the selling price should be comparable with other proteins which are available in the market. Next comes the competition with the commercial plant and animal proteins in terms of price and nutritional value. Also, the SCP should match the safety levels for human and animal food. SCP plant establishment requires large fermenters, efficient cooling system, which in turn require a large amount of capital. So, establishment of SCP plant in developing countries is now becoming expensive, where actually the need exists. This is a diagram which shows that how SCP is produced. It is produced from a substrate, and here two fermentation processes are shown. The dotted line are the semi-solid type fermentation and the continuous line are the submerged fermentation. These are the two pictures of large-scale fermenters. Next, we move on to some commercial processes through which SCP is produced. Number 1 is the ICI process. It is the process developed by Imperial Chemical Industries, and this process requires the bacteria Methylophilus methylotrophus, and the substrate used here is methanol. The SCP obtained by this process contains the protein of 71% and it is marketed as the name protein. It is used as a substitute of milk in feeding calves. This is the picture which shows how protein is manufactured. Now we move on to another process which is the RHM process. This is known as the RHM microprotein process and it is developed by the company Rank Hovis McDougall. This process uses the fungus Fusarium graminearum, and the substrate used here are glucose or starch, gaseous ammonia, salt and biotin. The SCP product has a meat-like texture which is marketed as microprotein and it is approved for human consumption. The figure below shows the packaged microprotein which is marketed under the name of Q-on, which says soy-free, meat-free chicken style pieces, like that meat-free meat chunks. This diagram shows how microprotein is cultured. Here we can see that starter cells are first harvested and then they are placed on certain types of substratums which are known as scaffolds. These scaffolds are actually biodegradable and are edible. The scaffold then containing the microorganism, that is the fungus, is then placed into the fermenter where it is allowed to grow. After the growth, the fungal biomass is harvested from the fermenter and then it is processed and vacuum packed and marketed as microprotein. The third process is the Bell Fromagaris process, which uses the yeast Cluveromyces merixinus and the substrate used here is whey. This process is a continuous aerobic type fermentation and the SCP produced by this process is of both feed and food grade. Now we come to the advantages of single cell protein. The advantages are that SCP give a high yield in a very short time. The SCP production uses substrates of very low cost and it also helps in environmental cleaning. The protein content in SCP is very high. Apart from proteins, other nutrients are also present like vitamins and minerals, so it can be used as food supplements. Microbial cultivation is much easier and is independent of climatic variations and seasonal changes. So, it is easier to cultivate microbes than plants and animals. Microbes can be genetically modified and the desired type of nutrient thus can be obtained, which is not possible in case of plants and animals in a very short time. Now we come to the disadvantages of single cell protein. Bacterial cells are small in size and low density, so the harvesting process are difficult and costly. In case of algal SCP, there is a high amount of cellulose and chlorophyll which is not digestible. They also accumulate some types of heavy metals which have proved to be harmful for humans and animals. Fungus and yeast SCP are deficient in sulfur containing amino acids. Some microbial SCP are high in nucleic acid content, which may result in kidney stone and gout. Also, high capital cost and facilities to maintain sterility makes the establishment of SCP plant expensive in developing countries. Now we come to the nutritional and safety evaluations. There are certain nutritional and safety evaluations which are: the chemical composition of SCP must be characterized in terms of nutrients like proteins, amino acid, nucleic acid, lipids, etc. Analysis of the substrate and toxic substances should be done thoroughly. Physical properties of the biomass used like density, particle size, texture, color should be thoroughly evaluated, and the nutritional value should be thoroughly evaluated on the target species. In case of products for human usage, evaluation should be done for longer periods using multi-stage processes. Now we come to the conclusion of this topic. SCP is now becoming popular as animal feed, but the use of human food is still controversial. SCP can be used as livestock feeding, which then reach to humans via animals. And further research and development is required to ensure the usage of microbial SCP as diet supplement in developing countries for both human and animals. However, it is noteworthy that many companies and organizations nowadays are becoming interested in the production and research of SCP. Thank you.